What I'll try and do in the next 25 minutes or so is, first of all, just say a few comments about what I think patients are looking for. I'll then share with you some thoughts around drug discovery. I believe the way we're doing drug discovery today, it's immensely inefficient. I'll share with you what I think is the biggest challenge. So I'm trying to come up with new drugs, not just for chronic diseases, but many rare diseases. And I'll share with you what I think is the biggest challenge. I'll share with you what we've been doing in Oxford and the impact we've had, what we've now started to do, what we're planning on doing, and then I'll sum up. So first of all, just thinking in terms of what do patients want? What do their carers want? What do relatives want? And maybe even what do payers want? What they want is novel medicines. They want effective medicines. They want affordable medicines. And they want them quickly. Now, in terms of, let me just throw a few numbers. And we've been talking about cancer. We just had two brilliant talks. You know, in the UK, in the next 12 months, 350,000 people will get diagnosed with cancer. That is 1,000 people every day. That is one person every 90 seconds. And half of us in this room, during our lifetime, will have a diagnosis of cancer. Across the world, in the next 12 months, 14 million people will be diagnosed with cancer. We desperately need novel treatments. I said we need effective treatments, and you might think, well, why is he saying that? Aren't the drugs that are out there effective? Well, there was a publication just a couple of years ago on solid tumors. So in this publication, they looked at 71 different treatments for solid tumors. They showed that those treatments produced an increase in progression-free survival of 2.5 months. An increase in overall survival of 2.1 months. And of those 71 drugs, only 30 produce clinically meaningful efficacy. Half of the drugs that are out there produced clinically meaningful efficacy. I said we need affordable drugs. So a publication last year, they looked at, in the year 2000, altogether, there were 69 drugs for the treatment of cancer. Of those 69, 50 were cytotoxic drugs. The average duration of treatment for those drugs was 181 days, so 6 months. The average cost of that treatment was £3,000. And in the year 2000, £3,000 was 20% of GDP per capita in the UK. That was 2000. They then did the same analysis in 2013, and they looked at another 63 drugs for cancer. The average duration of treatment now was 263 days. So it's gone up from 181 to 263. The average cost of that treatment was £35,000. So it's gone up from 3,000 to 35,000. And £35,000 equates to 140% of GDP per capita. So it's gone up from 20% to 140%. And that trend is continuing. We will not be able to afford these medicines. We need these medicines quickly. For patients, even a new medicine tomorrow is too late. They want them today. So what we need is novel medicines, we need effective medicines, we need affordable medicines, and we need them quickly. I said the way we're doing drug discovery today is incredibly inefficient. It's too costly, it's too risky, and it's too slow. Let's just put some numbers on that. Too costly. So Tufts analysis about a year ago, they said that the average cost of a new drug was now $2.6 billion. There was an analysis by Forbes about 2.5, 3 years ago, where they looked at a number of large pharmaceutical companies. They looked to see how much they spent on research and development over a finite period. They looked to see how many drugs those companies launched in that finite period, and divided one number by the other and came up with an average cost of a drug. In that analysis in 2013, the average cost of a new drug for AstraZeneca was close to $11 billion. Amgen, who are at the bottom of that table, the average cost for them was $3.5 billion. Even $3.5 billion is unsustainable. And bear in mind, many of those drugs that were launched in that analysis, they were Me Too's or new formulations. They were not completely novel treatments. And what we need is completely novel treatments. So it's too costly. It's also too risky. So another publication a couple of years ago, they looked at, in 2003, altogether there were 529 molecules in development for cancer. So they were either in phase 1 or phase 2 or phase 3. They then looked in 2013, a decade later, what had happened to those 529 molecules, and they found that 45 of them had made it to the market. Well, that's good. 95 were still in development, but 389 had been terminated. So we took 389 molecules into the clinic for the treatment of cancer and then terminated them. Imagine the resource that we've wasted on that. In that same analysis, they looked at, if a molecule is in phase 1, what's the probability it will get to the market? This is for cancer. The figure was 7.5%. Less than 1 in 10 molecules in phase 1 will get to the marketplace. In the same study, they looked at, if a molecule is in phase 3, what's the probability it will get to the market? The answer was 33%. So a molecule in phase 3, only 1 in 3 makes it to the market. The way we're doing drug discovery is too risky. It's also too slow. So my colleague Stefan Knapp published a paper in 2014 where he looked at, when did we initially get some data that inhibiting, for example, a particular kinase would be useful in cancer? And then how long did it take to take that idea into the clinic, into phase 1? The answer was between 6 years and 30 years, 3-0, 3 decades. 3 decades to take a new idea into the clinic. This process, the way we're doing drug discovery, is too costly, too risky, too slow. The biggest challenge we have is we do not know how to do target discovery. In each of us, there's more than 20,000 proteins. Each of those proteins could be a drug target. In the clinic, we've got hundreds of chronic diseases. Each of those chronic diseases is probably 20 different subtypes of disease, and we've got 7,000 rare diseases. Our ability today to say, if I take this novel protein and inhibit it, it's going to work in this disease or in this subset of patients, is appalling. This is why when we take new ideas into the clinic, 9 times out of 10 they fail. That's the biggest issue, target discovery. Let me share with you what we've done in Oxford. We've done 4 things. First of all, we've pooled resources. So I've had close to £60 million of investment from the Wellcome Trust. That has allowed us to leverage a lot of funding from industry. So we're currently working with 8 large pharmaceutical companies. They've each given us 5 million euros over a 5-year period. So we have 40 million euros of private funding. We're pooling resources to share risk. The second thing we've done, I only work on completely novel proteins. I'm not interested in proteins that everybody else has been working on for the past 3 decades. We only work on completely novel proteins, and we work on proteins or protein families actually that some people think are intractable. They're undruggable. We deliberately work on those because I believe my job as an academic is to drive innovation. So we work on these new proteins and we generate new high-quality reagents. We generate the purified human protein. I'm not interested in rats and mice. Purified human protein. We build assays for that protein. We work out the three-dimensional X-ray structure of that protein. We design small molecule inhibitors for that protein, and we also generate antibodies for that protein. All of these reagents are high quality, not because I'm smart or we're smart. They're high quality because we're working with this network of pharmaceutical companies. So we have access to their resources, their expertise, their infrastructure. Once we've generated these novel high-quality reagents, what we do, and these are starting points for drug discovery, what we do is we make them freely available. We give them away to anybody in academia, anybody in biotech, and anybody in pharma, because I believe that's the best thing I can do to facilitate science and therefore facilitate drug discovery. So you can imagine every academic who comes into my office wants to collaborate with us. They know I've got no secrets. I'll share all of our know-how, all of our expertise, all of our reagents. That transparency creates a lot of trust, which is great for collaboration, it's great for science, and it's great for drug discovery. So we're now working with a network of more than 300 academic labs all over the world, and I don't pay those labs anything. These labs take our reagents and they test them in their assays. It could be cancer, it could be inflammation, it could be metabolic disease, it could be neuropsychiatry, it could be a rare disease. They are crowdsourcing science on these new proteins using our novel tools. So we're crowdsourcing science. The fourth thing we do is that all of our data, all of our reagents, all the knowledge we generate, we share it with the world immediately. We share it as soon as we've generated it, even before we've started writing any manuscript, because we want to try and reduce all the duplication and wastage that's out there. At the moment in biomedical research, there are probably 20 groups across the world doing almost exactly the same thing, working on the same target, trying to generate the same sort of tools or the same sort of data, etc. We're trying to reduce that duplication and wastage. So that's what we've been doing. Pooling resources, driving innovation, crowdsourcing science, and releasing everything immediately. The impact of this has been phenomenal. So let me share with you one story. So 7, 8 years ago, as Rick knows, I started a project in Oxford to work on epigenetic proteins. I decided to work on epigenetics because I believe there's a rich seam of drug targets in these proteins. I believe we've overhyped the role of genetics in disease and completely underestimated the role of epigenetics. So epigenetics is all about how the environment affects gene transcription, etc. So we know diet, exercise, early life experiences, stress, injury, exposure to toxins have a massive effect on the initiation, the development and maintenance of many diseases, especially neuropsychiatric disease. So I started working on epigenetics. Now, the other reason for doing this was in epigenetics there were no tools available. Nobody had an inhibitor or an antibody or protein or assay, etc., etc. So we deliberately worked on that. Within epigenetics, there's a family of proteins called bromodomains, and I chose to work on this family and concentrate my resources on that. So in humans, there are 61 of these bromodomain proteins. When I started this project, many people, many scientists said, Chaz, you're mad. You'll never generate inhibitors for these proteins because they're undruggable. It's a protein-protein interaction. In 2010, working with GSK, we generated our first inhibitor. This was for a protein called BRD4, bromodomain 4. The name doesn't really matter. Once we generated this inhibitor, we thought, well, what are we going to do with it? So we looked in the literature, and there was some data suggesting that patients who have a fusion of this bromodomain protein with another protein called NUT, these patients have a very rare cancer called NUT midline carcinoma. So we decided to work with the world expert in NUT midline carcinoma, a guy named Jay Bradner in Harvard. Now, Jay has access to patients, he has access to patient material. So he took this inhibitor, he put it onto NUT midline carcinoma cells, and he showed that this compound reduced proliferation of those cells. So it stopped the cells multiplying. It also increased apoptosis, so it killed the cancer cell. He then took these patient-derived cancer cells and put them into a mouse. The cells multiplied, the tumor got bigger and bigger, and this compound stopped the tumor growing. When we got that data in 2010, Jay wanted to take this molecule straight into patients. The reason he wanted to take it into patients is because he knew that anybody that's diagnosed with nut midline carcinoma, within 3 to 6 months, they're dead. There is nothing out there that treats this cancer. That was in 2010. We published all of that data eventually in Nature in December 2010. Let me share with you now what's happened in the last 5 years. That molecule we generated, we have now shared, given away for free, to more than 1,000 labs across the world. These labs have taken that molecule and tested it in a whole range of assays. They've now shown that it works in a whole range of cancers. There's data in fibrosis, in COPD, in cardiac hypertrophy, in sepsis, and there's even a publication in male contraception. As a consequence of that one molecule, there are now more than 300 publications on that target in 5 years. What's now happened is that many of our pharmaceutical partners have started their own proprietary program to generate their own molecule. And today, there are 6 companies with 6 different molecules undergoing 14 clinical studies. On top of that, Jay Bradner, 5 years ago, managed to secure $15 million of VC funding to set up a biotech in Boston to take that target all the way into the clinic. And a month ago, I was just saying to Rick and Louise that a month ago, Roche bought that company for $535 million. So what have we done? One molecule, high quality, freely available to the world, that's catalyzed science, catalyzed drug discovery, catalyzed the creation of new companies, and catalyzed the creation of new jobs. We've now generated 37 of these molecules, and all of them are freely available to the world. I've just shared a story with one of them. So what are we doing now? So that's the impact. What are we doing now? So we've just started a new project funded by the Wellcome Trust. So what we're now going to do is we're working with clinicians, disease experts, geneticists across the world, both in academia and industry. We're going to take high priority genes that are coming out of these large-scale genetic studies, and then we're going to do what we're good at. What we're good at is generating the protein, assays, structures, chemical starting points. This package of reagents we've called a target enabling package, and of course it'll be high quality and we will share it with the world. Because I believe, the best starting point it is to start from human disease, human genetics, etc. The second thing we're doing, some of you know I'm not a fan of animal models and I'm not a fan of cell lines. I think largely they're misleading. I have seen too many things work in animal models and I take them into the clinic and they do nothing. So what I'm more interested in doing, sitting in Oxford, I'm surrounded by hospitals, clinicians, access to patients, access to patient material. I want to test my inhibitors on primary human cells or tissues. So we're focusing on inflammation, and so we're creating this platform of primary human cells from healthy individuals, but also patients, and then we're going to test our molecules in those. I think that's a much better way to come up with new targets for drug discovery. The third thing, we've just got some funding from the Brazilian government. So a couple of my guys have gone out to Brazil now to set up a group in Brazil. So you're probably aware in humans there are 518 kinases. If you look at all the literature's on about 50 of those kinases. If you look at the patents, they're on about 30 of those kinases. There's probably about 300 kinases that we've not even touched. So what we're going to do is to generate novel inhibitors for those kinases and again, make those freely available to the world. That's what we're doing in Brazil. Last year, we also got funding from Alzheimer's Research UK to set up a dementia institute in Oxford. This is a disease, it's a crisis, frankly. If we don't have a drug for Alzheimer's in the next 15 years, that one disease is going to financially cripple many societies. So what I'm going to do is I'm going to open up completely new areas of biology. I don't care about amyloid and tau that everybody's been working on for 3 decades. We want to completely focus on different pathways, different targets that could lead to new drugs for dementia. And of course, all the tools, all the data that I generate, I will share with the world. I won't be keeping it secret. The other thing that we've just started doing is building more and more links with patient organizations, because patient organizations are desperate for new treatments. Of course, they have access to patients, they can access patient material, so we're developing collaborations with them. We've just formed a linkage with Myeloma UK, the Huntington's Foundation, and just recently the Brain Tumor Charity as well. Those are some of the things that we're doing pre-competitively, freely available. What we also managed to do a couple of years ago was get some money from the UK government. So what's been happening over the past few years, we've been generating all of these tools, and then our academic network's been generating lots of new biology and new disease understanding. And then what's been happening is that many of our academic collaborators in Boston or San Francisco or Shanghai, they've been setting up new biotechs. And of course, the UK government wants to see those set up in the UK. And of course, we want to do that in Oxford. So they gave us £11 million to build a bioescalator in Oxford to make this happen. So those are some of the things that we've started. What I'm proposing to do is the following, and this is our most ambitious project. I believe target validation happens in patients. It doesn't happen in animals. It doesn't happen in cell lines. We need to get some of these new targets into patients, into the hands of people like Rick and many of you as quickly as we can, because that's where we really find out if a target's going to do something. At the moment, what's happening is that there's maybe 20 companies, they work on the same target in parallel, in secret. They spend 5, 6, 7 years coming up with their own proprietary molecule. They take it into the clinic, and if one of them fails, the other 19 fails as well. That is a tragic waste of money. It's a waste of people's careers, but it's also a waste of patience. Ethically and morally, this is wrong. So my argument is we should do that clinical experiment. Let's do it. Let's pool our resources. Let's do it once. Let's do it well. Let's share the data. Let's identify the 9 in 10 that's garbage and the 1 in 10 that has the potential to be a new drug. Once we've identified that 1 in 10, then companies like GSK, Pfizer, whatever, they can come up with a proprietary molecule. At least they know it's clinically validated, it's been de-risked, they can come up with a high quality molecule, do the big clinical studies, take it to the marketplace. It's good for patients, it's good for industry, it's good for society. So to sum up, I believe in early discovery, there is too much competition, there's too much secrecy, it's leading to too much duplication and too much wastage. What we're trying to do is to create a completely new ecosystem involving lots of clinicians, lots of academics, lots of pharma companies. We're also working with a number of biotechs and CROs, which I haven't mentioned, and now we're working with several patient organizations. We're funded by funds from charitable organizations, philanthropy, private organizations, and government funded. And together we're trying to come up with new targets for drug discovery. I believe that if we put this in place, we will generate more novel medicines more quickly. And I hope, ladies and gentlemen, that these will also be more affordable. Thank you very much.